Okay, I think we can start. I don't sure if yeah, we have 49 participants. So welcome everybody. I am Joseph WGLP. I'll be chairing these two sessions on the data track. That is about, it's difficult to define. It's a very heterogeneous thing, but we can perhaps call it data science in a more general view. We have extremely complicated statistics for me, also some machine learning, some traditional prediction schemas, but well, there's no single definition for that. Hopefully, it's going to be very useful sessions. We're split into sessions. One is now from the next hour, and we have a second session from 12 on in our time. And we have 4 speakers now, 5 speakers later on, so it's quite tight timing. We expect somehow 12, 13 minutes for presentation. So I expect from the speakers to be as much as attached to the time. I will try to warn you around 10, 11 minutes presentation. So we have 1 more minute and we have some time for questions. So without much more delay, I think I will introduce the first speakers. If I'm not wrong, is Donji Lee from the Yanjun University in South Korea. Is that correct? And I don't have the title right now, so my fault. Too many screens, I'm afraid. Yeah, it's about the dementia key gene identification using multi-layer SMP gene disease network. So I think, Donji, you have the floor and go ahead. Thank you.
Okay, thank you for introducing me. Good morning and good afternoon and good evening. I am Donggi Lee from Yanjun University in South Korea. First of all, I'd like to thank the organizers of ECCB. It is very big honor for me. And also I want to express my appreciation to all of my colleagues, collaborators, and especially my advisor, Professor Shin. So the topic of our research is identifying key genes for dementia with multi-layered network. So this is an abstract of the paper. Let me skip it. I will talk with the following order. There are huge achievements in computational biology of decreasing cost, time, and efforts in the case. It gives us advanced approaches to find key genes of diseases. The key genes play a critical or central role in diseases, so determining key genes is very helpful and important for diagnosis or treatment. Specifically, several types of dementia are known to be affected by genetic factors, and APOE gene is a very famous key gene for Alzheimer's disease. In addition, various genes are observed as associated with dementia in recent years. These discoveries were conducted through GWAS by selecting specific SNPs and targeting genes that contain selected SNPs. So the SNP is the most common form of DNA polymorphism due to the difference in single base pair variation among individuals. So it can use as a marker for a physical or genetic mapping. I'm sorry. These SNPs appear as some locations in a gene and affect the gene. And then the presence or absence or mutation of a gene is related to a disease. So we consider these relationships for identifying key genes of disease, especially dementia. In this study, the mutual systematic relationships are reflected as hierarchically structured networks. In the network, there are intra-relations within each SNP, gene, and disease layer, and inter-relations between layers are also included. And then we find specific genes that are yet unknown but may play a significant role in dementia. Such genes are defined as candidate key genes. So this is a brief overview of the method. First, a multi-layered network is constructed with public data and disease-specific data. With the network, we apply a machine learning algorithm, then the scoring process is conducted. Finally, candidate key genes for dementia is predicted. For the method of multi-layered network construction, I will introduce each network one by one. Firstly, in the disease network, edge weights are calculated by cosine similarity from the disease protein or gene relation. In the gene network, the edges represent correlation scores. These scores are pre-calculated through below 7 information and formula from the string database. The string database is a well-known database for the gene or protein network, and it is very useful, so we utilize it. And next one is a SNP network. Like disease or genes, there can be complex interaction between SNPs. So for the edges in the SNP network, we employ epistasis test in Plink. The epistasis test is performed using the following logistic regression model that classifies case and control groups from a pair of SNPs. From the test, the interaction between SNPs is computed based on the odds ratio from the model. If this odds ratio has a value of 1, then it implies that there is no interaction between two SNPs. However, when the value goes away from 1, it means that there is a stronger interaction between the two SNPs. To reflect this, the following transformation function, T of x, is applied to the odds ratio to convert the value into similarity, and the resulting value is used as edge weights. So disease genes and SNP networks are separated single networks at the same levels. But however, interrelations depict relationships between two nodes in different levels. Interrelations are based on information of SNPs belonging to specific genes and genes affecting specific diseases. Interrelation weights take binary values of 1 if a relation exists between two nodes and 0 otherwise. The next section is about the machine learning algorithm. We employ a graph-based semi-supervised learning algorithm, which is useful when data is represented as a graph or network. The formula of the SSR algorithm is like this, and let me skip the details. So why we use the SSR is that the algorithm is very powerful if there are few labeled data. We have tremendous number of SNPs, genes, and diseases in the network, but the number of labels that are related to dementia is very small. Also, the multi-layered network is represented by a huge matrix. This huge matrix can lead to intractable calculation for a basic SSR. So we employ an SSR algorithm specialized for a multi-layered structure. This is a summarization of the algorithm. It can boil down high computational complexity. And also, let me skip the details. By the hierarchical structure, we can uncover an unknown knowledge through interrelations. For example, many diseases are disconnected in a single network on the left side, but with interrelation, those are connected based on protein-protein interaction information on the right side. That is why we use a multi-layered network. Furthermore, label information is necessary for the prediction process. In the disease network, the disease dementia is set as label, and the genes that are already known to be related to dementia are assigned to label in the gene network. For the SNP network, SNPs with a high association with dementia are set with label. The associated SNPs are selected from the following statistical tests. The lowest p-value is set as the representative p-value. So SNPs with p-value below a threshold delta are set as labels. Finally, after setting the label information, the SSL algorithm is applied. The output values of the algorithm are represented as scores with following normalization. The score implies that higher scores of genes have higher possibilities of candidate key genes for dementia. And the next is experiments. Firstly, we collected data as public and disease-specific from multiple sources. In the first row, patient genome information is dementia-specific data, and the others are public data. For dementia-specific data, we collected human biospecimens from the chronic cerebrovascular disease-oriented biobank in Korea. There are about 830,000 SNPs of 186 patients with various types of pathologies. From the types of dementia, we focused on Alzheimer's disease and vascular dementia, which are the most common dementia. Thus, there are a control group with normal or MCI patients and 3 groups for AD, VD, and both simultaneously. This result shows construction of multi-layered network, and disease and gene networks are commonly used irrespective of patient groups, and each SNP is based on the groups respectively. And this is the number of interrelations between layers, and labels are assigned according to the disease of each group. The next result is about the performance. The performance of the proposed method was compared with that of the gene disease two-layer network without the SNP network. This was conducted to measure the effect of the SNP information on predictive performance. The experiment was set as on the screen. And the result shows the better performance in all groups. More specifically, in the AD group, the performance improved remarkably and showed a stronger stability. And for the VD group, the increase of AUC was relatively lower, but the stability improved. This result can be explained pathologically. The vascular dementia is caused by external factors rather than genetic factors, and the proposed method searches for genes that cause dementia, so the result can be reasonable. And the AD and VD group shows compromised results because it contains a mixture of both genetic and non-genetic factors. After validating the performance of the proposed method, we applied it to key gene identification. This shows a summary of the overall process to AD. So this figure depicts the result. In the figure, the gray nodes are already known genes to be related to AD, and the red nodes are newly recommended candidate key genes through the proposed method. We couldn't verify all of the candidate genes, but some of them were verified to have associations with AD by retrieving PubMed literature. Such genes are highlighted in red. And for example, MYRIP and DDR1 are reported that are related to AD or neurodegeneration in some papers. And this table shows more candidate genes with verifications. And in conclusion, we propose a method to construct a multi-layer network and recommend key genes for dementia using a machine learning algorithm. The network reflects the effects of mutual interactions, and the results show the outstanding performance with SNP information. So we focused on the disease dementia in this study, but the method is flexible to other diseases. Also, we think that the method can be extended to personalized gene or SNP identifier. It is last page of my presentation. Of course, there are some limitations of this study. The resulting network may be biased to the current data set, which has been collected from only one biobank. To resolve this issue, diverse public data, such as ADNI or UK Biobank, and cohort study can be used to avoid. This will be our future work. And this is end of my presentation. Thank you for attention.
Thank you, Dongi. Thank you for sticking to the time. I remind all the participants that there is a place to put questions that is how is it called, live Q&A. And eventually you cannot talk, but I will try to read the questions in a meaningful way. We have one. The Pau is asking if that method accepts weighted networks.
Yes, the method accepts weighted network. I have not explained in detail, but the constructed network itself has weighted edges. Yeah. So my answer is yes.
No more questions for now. I have. You said that this is easy to generalize to other diseases, to other environments. How easy is that? I mean, which is the kind of information you need to collect? How much information you need? I understand that the protein-protein directions will not be the same,
yeah,
because string is general, but you have a lot. You need a lot from the SNPs and a lot from the diseases. Yeah. I think is that.
Because disease network and gene network is based on public data, and only SNP network is based on the disease-specific data. So if we have other disease-specific data, like genomic data, then we can construct a new SNP network for that disease. Then we can use it flexible, I think.
Okay, we start having questions. That's growing. So from Laura, how the method copes with the SNP located in non-coding regions? In particular, how do you deal with those SNPs for the SNP-gen relationship? This is important because the SNP identified by GWAS are mostly non-coding, and it's challenging to map them to genes.
In the proposed method, SNP gene relation is SNP and gene relation has just the binary value if there are relationships on DB SNP. So we didn't quantification of that, but I'm not sure it is. It can be an answer or non-coding regions. I just used from the DB SNP database, so it may be a simplification.
this question is other people in the chat is asking the same thing. So I understand that you are linking genes in a more wide way, and as Laura was saying, non-coding regions are becoming extremely present now because we are no longer using traditional SNPs, but also exomes and all these things, and even whole genomes.
I'm sorry, I didn't understand about the coding regions.
No, non-coding regions, regulatory regions. Anyway, just go. That's the last question because this is getting later on. We have a comment from Iker. Nice talk. How much could this method be adapted for diseases with a lower number of associations, for example, rare diseases?
I think I didn't think about rare diseases, so I have to concern about that with sometimes. I will answer on the live Q&A on chatting.
Okay, I think perhaps we are getting late, so perhaps we can you can just keep discussing this offline with Laura and Iker. Thank you, Dongi, and we'll pass to the next speaker. Thank you. And the next speaker, if I can find the program, sorry, my screen is very small, is Thomas Gumbsch. My pronunciation is, sorry about that, from Zurich, from the ETH, that is going to talk about enhancing statistical power in temporal biomarker discovery through representative shapelet mining. Thomas, floor is yours.
Okay. Good morning. Thank you for the introduction. In this talk, you will learn about representative shapelets with the focus on temporal biomarker discovery. I cannot move the, oh yeah, now. Okay. First, we introduce the problem of finding a time series subsequence that can be used as a biomarker. Then the method is introduced before discussing shapelets that we find to be associated to in-ICU mortality on the EICU dataset. Assume the following setup. We have patients in the ICU that exhibit a certain trajectory. In our dataset, we have access to the complete trajectory. So we have vital signs, drugs, infusions, ICD codes, and so on. Some of these patients die, and we want to find a biomarker that is associated to in-ICU mortality. Traditionally, a biomarker is a time-independent variable, but here we cannot find such a variable or threshold. What distinguishes the two patient groups, however, is that all patients that die exhibit a certain temporal pattern in their blood pressure trajectory shown in red. We call this a shapelet, and we would like to find those with three specific requirements. First, they have to be physiologically reasonable. We can be sure that they are if they are subsequences that occur in the data. Two, since clinicians are interested in p-values, we need to be certain that the patterns that we find are statistically significant. That means we have to correct for all the hypotheses that we test. So in this case, we have to correct for all the candidate shapelets. Three, since a significance threshold does not impose any structural condition on the set of shapelets, we also require the set of shapelets to be of manageable size and structurally diverse to be interpretable by humans. First, we note that traditional time series analysis methods deal with meta-quantities or non-local information that cannot be directly related to a pattern that can be observed. The only approach extracting subsequences occurring in the data that are related to a phenotype are shapelets. In the original shapelet publication, candidate shapelets were extracted with a sliding window method. The set of candidates contains all subsequences of the dataset. These subsequences are then ranked on how well they predict the correct class label of the time series. A prediction from a shapelet comes from the difference to the best fit to the time series. That means shapelets are here used as a feature extraction approach. In 2018, our lab extended this approach to biomarker discovery and to statistically validating the shapelets. To rank the shapelets, we employ a chi-square test and determine the significant subset of these by using the Tarone multiple hypothesis correction. The Tarone correction is less conservative than a Bonferroni correction because it deems a proportion of the patterns to be untestable, depending on how often they occur in the data. Note that the Tarone threshold is defined recursively. That means we have to loop over all shapelets. The S3M approach is the only in the literature to fulfill the first two properties, and we have used it in a variety of domains. So for instance, we used it for prediction of circulatory failure in the intensive care unit. What we had to do each time, however, is a post-processing of the significant shapelets because the set of shapelets are redundant and contain flat subsequences. A flat subsequence corresponds to a fixed threshold, and the redundancy originates from the original shapelet candidates being overlapping subsequences. A big open problem is therefore to extend our approach to also contain a selection of representative and significant shapelets. The shapelet selection approach that we propose is solving a submodular objective function. Submodularity is the set analog of convexity, and solving a submodular objective can be accomplished with a greedy algorithm. Our submodular objective function consists of three parts. The bottom equation rewards coverage of the full set S with the representative set R. This is called the facility location. The sum redundancy function is the middle equation and rewards dissimilarity of the elements in the representative set R. The last term rewards small cardinality of the set R and is the last term in the top equation, I'm sorry. The three objectives are mixed with a trade-off parameter lambda. It is the same objective as in Liebrecht et al. From 2018 because it is the only approach in the literature that discussed submodular subset selection on sequences. We searched for representatives in time series subsequences. They developed their approach for protein sequence data, which is close enough for our purpose. So essentially, we search for representatives by trading off similarity to non-representative items with similarity to other representative items. This is now an additional step for our shapelet selection pipeline, where we select representative shapelets. Note that the alternative to selection of a representative subset is clustering. However, clustering is a problem for unequal length shapelets because the underlying distance measure is not guaranteed to fulfill the triangle inequality. In contrast, representative subset selection extracts individual items chosen to represent a larger set, which has fewer requirements. For this approach, not even symmetry is required from the underlying distance measure. Compared to the Liebrecht approach, we changed the optimization algorithm because the greedy and their proposed bidirectional greedy algorithm are quadratic in runtime and therefore computationally too challenging for processing all subsequences in the data. We propose an iterative version of the bidirectional greedy algorithm that approximates the optimal solution in linear time. In the figure, you can see in red the prototype shapelets that we use in our simulations. In the simulation dataset, these are repeated 10 times each. And the columns show how many of the representatives are extracted by each algorithm. Not only in this simulation, but also on other larger scale experiments, we see that the iterative version, here big, approximates the optimal solution similarly well as the other two algorithms. Remember that we are confined to extending the S3M approach for our purpose because we are interested in statistically validating the shapelets. It is not a trivial question where the shapelet selection step should be placed in the pipeline. What is shown here is pre-processing shapelet candidates. So after generation, we find a representative subset of the shapelet candidates before ranking and significance testing. We project that this approach is less sensitive because the representatives are selected without knowledge of the class label. In a post-processing approach, we find a representative subset of the significant shapelets. In this case, the significance threshold is expected to be too conservative because the non-representative shapelets are discarded. The proposed S5M method deems non-representatives as non-testable and combines significance testing and representative selection simultaneously within the Turon method. Note that this can be accomplished because we use an iterative solver for the submodular objective function, where the full set of representative shapelets does not need to be known in advance. What we have just discussed can be confirmed on simulation and real-world data sets. The simulation on the left contains time series where the prototype shapelets have been inserted. S5M has superior distribution of F1 scores on a variety of data sets compared to its two-step alternatives. In these data sets, we are varying the sample size and the noise. On the right-hand side, shapelets associated to sepsis in the ICU on MIMIC3 are extracted. It is the same setup as with the S3M publication. First of all, we find that S3M give too many shapelets. No one is able to process 100,000 shapelets in the blood pressure setting. When comparing S5M to a post-processing, where we select representatives after assessing significance, we find that S5M is of greater power, yielding slightly more shapelets that are also structurally more diverse. The below and above columns are interesting thresholds from the literature, and if a shapelet reaches into that region, it is potentially more interesting than a shapelet that lies within a normal range. The eICU data contains 200,000 ICU states from multiple sites in the US. For these patients, we extract the SOFA score, which is a proxy for severity of illness. The SOFA score is a composite score containing clinical variables from different organ systems. Plotted on the left is the mortality depending on the maximum SOFA score during the first 4 hours. And as expected, the mortality increases with increasing maximum SOFA score. The mortality also plateaus at 10 with around 30% mortality. The question is, when only looking at patients with maximum SOFA score greater or equal to 10, can we find representative and significant shapelets that are associated to in ICU mortality? And the answer is yes. Note that we search for shapelet of length between 2 hours and 4 hours. The first 2 shapelets are associated to not dying in the ICU. The last 3 shapelets are associated to dying in the ICU. Clinically, they make sense.
Thomas, sorry, is one more minute.
Yes, this is my second to last slide.
Okay, fine.
Okay, so clinically, the 5 shapelets make sense. The first 2 show a decreasing SOFA score, which shows a patient is improving, and the last 3 shapelets show a increasing or high SOFA score, which means that the patient is in a bad state. We have 2 consequences. So the first consequence is that in a clinical setting, usually the SOFA score is computed every day. We advocate to compute it hourly and from the electronic health record directly for the clinician in the bedside. And we aim to use this as possible biomarkers. So one last sentence to summarize, S5M is the only method that finds a set of temporal biomarkers that are occurring in the data, that are statistically validated, and that are interpretable. Thank you.
Thank you, Thomas. The time is a few minutes for questions. I don't have questions right now, or at least I don't find them. Any questions from the audience? I understand that this is a tough issue. I have a general, philosophical question, I would say. This is general for time evolution events. You mean, this is a clear use case, what you're explaining, but my question is if that is general for anything that is a time series set of data. Can be used in a similar way, or is something that is very focused to this kind of data?
We have only used it for health-related data. So we have used it on the ICU data sets like MIMIC3 or eICU. We have used it from ICU data in Switzerland. We have not used it for regularly sampled data. So we developed this approach because we want to find patterns that are occurring in the data. So we want to find shapes of the blood pressure trajectory. We want to find shapes in the heart rate that are of diagnostic relevance. And this was our goal. So we have only applied it to health data yet. But of course, you can extend it to other domains.
No, that's interesting point. I work in simulation, so it's a bit more. It's a very different thing. But I'm still waiting for questions from the people. But I can keep talking just one more minute. If you mix different types of input data, you said blood pressure. You mean you need a similar rate on that thing, or something can be, I would say, measured once a day, other every 2 hours, or every 2 minutes, and that works samely, or you need some kind of synchronization?
We don't need that synchronization. If I understand you correctly, you mean how does data which is sampled only every day related to data which is sampled every 5 minutes? And this approach that we propose is a univariate approach. So we look at each channel independently. And of course, the question is, and this is also our work now, our future work, how to extend it to multivariate data. So maybe we can find also shapelets in blood pressure and lab tests, for instance. Was this your question? I don't know if I get it correctly.
Yeah, that was it.
Okay.
I think, because we thank you, Thomas. I think that we don't have much questions from the audience, and we have getting late. So we'll pass the floor to the next speaker. That
Thank you.
Understand. Thank you, Thomas. I understand that is Tamim Abdelaal from Delft University in the Netherlands, that is going to talk about the Schnell scalable clustering high-dimensional single-cell data. Tamim, the floor is yours.
Thank you very much. Welcome, everybody. Today I'm going to explain to you, to use quickly our new clustering tool for high-dimensional single-cell data, as we call it, Schnell. So let me dive directly into the topic. So the main question here we're trying to answer is, in single-cell studies, how do we usually identify the different cell populations you have in your pool of cells? So basically, starting from the left side, where you have your high-dimensional single-cell data, where you measure, let's say, protein or gene expression, and you're trying to go to the right side, where you define the different cell population that you see. And the main tool used in that case is clustering. So you cluster your data, you define different groups, and then you want to put some biological interpretation on top of that. So you can look, for instance, for marker genes. And then depending on these marker genes, you can then annotate your clusters. Like, for instance, this red cluster can be annotated as B cells, because we can see that it does express the marker gene C19, which is a marker gene for B cells. So as we may all know, that the single cell data over the last decade have been exponentially growing in the number of cells that is measured every month. So starting from 2009, where we have very, very few cells measured, now we have millions of cells measured every month, not even across different studies, but here I can even point out one experiment. So here I'm showing transcriptomics, so single cell RNA sequencing datasets. And this is a study from 2019, where they measured the mouse organogenesis. And in a single study, we had closer to 2 million cells. And if I consider a different single cell technology, something like the cytometry, for example, we can easily go up and scale up to 9 or 10 millions in one experiment. So this is an experiment of colorectal cancer, where they measured almost 9 million cells in one experiment. So moving back to the clustering, so that of course include challenges on top of the clustering and the data analysis in general. So the most widely used and the most trusted clustering used in single cell data, we can say that it's a graph-based clustering, something like the Louvain community detection, where you do build a neighborhood graph of your data and then try to define the different groups or the different communities in your graph. But referring back to the previous slide, where we know that now the amount of the size of the cells and the amount of data we measure is actually exploding, then the graph clustering have a clear scalability issue. So when you go above, let's say, 1 million cells, then computationally wise, it's not feasible anymore to apply the graph clustering and get a reasonable clustering. So of course, one way is to do something like random downsampling, but then you don't know what exactly you're throwing away, which we're trying to avoid. So that is mainly the main question we're trying to solve here, how to make the graph clustering more scalable to the size of data sets we have nowadays. And to answer this question, I'm going to take you to something slightly different, but a bit related, which is the visualization using the TSNE, which is one of the most popular nonlinear tools used for visualizing the single cell data. So here I'm showing the hierarchical SNE, or the HSNE for short, which I can call that this is a scalable TSNE. So the HSNE was found like in 2016 by colleagues in my group, and it was actually a solution for the scalability of the TSNE, which is very similar to the scalability of the graph clustering we're facing now. So also if you try to apply TSNE for more, let's say, than 1 or 2 million cells, then computationally this becomes very expensive, and also the map that you end up with is not visually anymore, so you cannot really see any structure. So the HSNE was introduced as a solution for that, and the H stands for hierarchical. So you build the hierarchy and you try to visualize this one. And I will introduce you quickly how the HSNE works. So let's assume that this is the pool of cells that you have. So this is all the cells. So instead of having one TSNE embedding, when you see all the cells, you can just divide this into hierarchy and visualize fewer cells. And the hierarchy is built from the data level, and then you go up in building hierarchy with less and less cells. And you aggregate the cells by what we call here landmarks, showing here as white points. And you select these landmarks based on the local connectivity in the neighborhood graph. So on the data level, you do build the neighborhood graph, and you select the cells which are highly connected in this graph as candidate landmarks for the consequent scales in the hierarchy. And one of the most important concepts in the HSNE, as we call it, the AOI, or the area of influence, showing here by these gray regions, that is actually saving the related information between a landmark on a scale and the data points on the previous scale. So in other words, which landmarks does represent which cell points or which points in the original graph on the previous scale. So for instance, this region over here is representing, I'm not sure if you can see my pointer, but I hope so. So this region is representing that this landmark can represent these 3 cells and so on. And one of the most important features of the HSNE that when we define the similarity above the data level, we do not rebuild a neighborhood graph on top of the landmarks, because then we lose the entire structure of the data. So the similarity beyond the data level is defined based on the overlap between the area of influences. So when 2 cells or 2 landmarks overlap in their area of influence, then they can be connected in the graph, and in that way you can retain the nonlinear structure that you have in your data throughout the whole graph. And as I mentioned in the beginning, this was actually used for the visualization. So you can then use these landmarks and only visualize. So we decided to use the same, let's say the same trick to scale up the graph clustering. So let's say that now I have this high dimensional data on this spiral manifold, where I have a lot of cells, and then I apply the HSNE, I build the hierarchy here for this illustration. I'm showing 2 scales. So you have fewer landmarks on scale 1 and more and less on scale 2. And you have the information encoded in the area of influence. And then let's say I can decide, okay, now I can apply the graph clustering on scale 2, which I have fewer and fewer cells. So computationally, it becomes feasible to apply the graph clustering in that sense. And then through the information encoded in the area of influence, it can actually flow back the information all the way back to the full data clustering. And then I have all the cluster labels flowed back to the data. So in the next few slides, I will show you some of the results we actually obtained when applying Schnell. So first of all, we applied Schnell on the popular machine learning dataset MNIST, which contains handwritten digits, images of handwritten digits from 0 to 9. And we applied Schnell, we created a hierarchy of three scales. And as you can see here in this table, that whenever you go into the hierarchy, you have actually less and less points. And the number of clusters, the original number of clusters, of course, is 10 because we have 10 digits. And in all 3 scales, we obtained something which is close to the 10, so from 13 to 9. And we evaluated this quantitatively using 3 scores. So the RI, the adjustment index, which is measuring the similarity between the original groups and the clustering we obtained with Schnell. The HS stands for homogeneity score, which is basically measuring the pureness of each cluster. So how pure each cluster is in terms of the groups that it contains from the ground truth groups. And the completeness score, CS, is basically measuring slightly the opposite. So basically if one complete ground truth group is obtained completely in one cluster. And the most important thing to note here that all the 3 scores ranges from 0 to 1, and whatever it's higher, closer to 1, it's actually better. And as we can see here in all the 3 scales, we obtained relatively high scores, which is all above 0.8. And interestingly, if I have a closer look at scale 1, for instance, so we see that we have 13 clusters. If I look at the contingency matrix between the original digits or the original groups that we see on the rows and the 13 clusters we obtained as on the columns, we can see that in most cases, there is one-to-one correspondence between the original ground truth tables and the clusters we obtained, except for cluster 5 that we see a division in 2 groups, and cluster 1 that we see a division in 3 groups. And if we have a deeper look, for instance, at the 3 clusters of digit 1, it's interesting to see that Schnell is finding relatively meaningful substructure. So if I look at the centroid of each of these clusters, I can see that, for instance, one is showing the handwritten ones, which is completely vertical. One is showing the tilted one with 45 degrees, and the other one is showing something in between. And next, we also applied Schnell on 3 benchmarks datasets for cytometry datasets. So the ML, BBMC, and Panorama, which are popular benchmarks cytometry datasets. And we compared Schnell with also the state-of-the-art clustering tools for cytometry data, so FlowSum, Phenograph, and XShift. And here I'm showing just the T-SNEs with the original and the clusters partitioning that we can see. So as you can see, for instance, FlowSum is just aggregating into very large clusters, so it doesn't really show the original manual annotations. Phenograph and Schnell, they show relatively close structure in the division, while XShift is actually having problems in defining the correct boundaries. And in terms of quantitative evaluation, we actually used the adjusted rand index because we know the ground truth table for the dataset. So we used the adjusted rand index as a quantitative evaluation. And we obtained a better performance for Schnell for both the ML and the Panorama dataset, and secondly scored with a slightly lower performance than Phenograph for the BBMC dataset. And it's not very surprising because Schnell and Phenograph, both of them are actually based on Louvain, so they are both using the same clustering technique. But if we look at the timing-wise, which is the scalability that we're trying to illustrate here, so Schnell was at least order of magnitude faster than Phenograph on all the datasets. Here I'm showing the data size on the x-axis and the time on the vertical axis. And this dataset, the largest one we tested, this HMIS dataset that contains actually 3.5 million cells, where we could actually obtain a clustering using Schnell, but Phenograph was not able to obtain a clustering after a week of runtime. And the last piece of results I want to show here again to extremely show the scalability of Schnell to large datasets.
Tamim, 1 more minute, please.
Yes. Yeah. So we were able for the first time to apply a single clustering of the entire Phenograph data. This is data was published with the original Phenograph clustering tool. So it obtained 17.2 million cells in the original study. This was actually clustered using Phenograph, but per individual. So it wasn't pooled. So we pulled all the individuals together, and we created a 6 hierarchy using the HSNE, and we clustered. So we skipped the first one because the amount of cells is still quite large. And we clustered the rest of the scales. And here I'm showing the cluster frequencies. So what you see here is on the rows, these are the healthy patients and the leukemia patients they have in the study. And on the columns, these are the 47 clusters at scale 5. And what the color gradient is showing is basically the distribution of these 47 clusters across the patients. So in that case, we didn't have a ground truth, so we cannot really quantitatively evaluate. But at least what we can see that we do see homogeneous structure across the healthy patients as expected, but rather heterogeneous structure across the leukemia patients. So in conclusion, I hope I was able to convince you that our new clustering tool, Schnell, represent a reliable and automated clustering for high-dimensional single-cell data. And using the HSNE, we were able to scale up the graph clustering to tens of millions of cells. And one of the benefits also of Schnell that we can apply this clustering at different scales, which can actually show a different level of details as the user can ask for. So finally, Schnell is now available as a Python package through our GitHub, as we call it Schnell Py. And we also have another version, we call it Schnell R, which is still, it's also available through GitHub, but it is still under development. Yeah. Thank you very much. I would like to thank all my collaborators, especially Paul Reddit, who did most of the work, and our funders, and you for your attention. Thank you.
Thank you, Tamim. We don't have questions in the chat. At least I don't see them. I have a quick one. How much biased is your first hierarchy? So is a blind thing or is something that you have to decide, look into the data? Because actually you are doing a kind of playcastering to make easier the full thing. So we're already doing something at the beginning that may be biased.
So basically, the full hierarchy is data driven. So you start by building a neighborhood graph for all the cells, and then you just select the cells which basically, let's say, have more dense neighborhood in the graph. So this is usually applied through, technically this is selected through random walks. So I apply different random walks in the graph, and then whenever you pass by a cell or by point more times than the rest, then this is a more candidate to be a landmark. And then you move on to the next scale and you apply the same thing. So the full hierarchy building is data driven. The only thing that the user can select in the beginning is the number of scales. So how many scales do you want to create? And we do have a recommendation based on that is to use slightly an equation, which is log 10 of the number of cells divided by 100. So that gives you an estimation of how many scales should you actually ask from the HSNE.
Thank you. We don't have questions in the chat, so I think it's really getting late. So thank you, Tamim. I will just pass the floor to Yanjun Kai or Ki. Sorry for that. That is from the University of Virginia, and she's talking about the FAST-SQ fast sequence analysis with gap string kernels.
All right. Hello. Okay. Great. Hello, everyone. So this is Yanjun Qi from University of Virginia, Department of Computer Science. So today my talk title is FAST-SK, fast sequence prediction analysis with gapped string kernels. And we have provided our tool via this link called github.com qdata FAST-SK. All right, so next page. Sorry.
I think you can pass yourself.
Yeah, can you help me to go to the next page? I think somehow maybe okay, let me see why this. Okay, I got it, I think. So can the control room just help me go to the next page, please? No. Okay,
Nice.
Can I share my screen? Like just show my slides?
Yeah, sure. I think we can share your screen.
That's actually easier. So it's not allow me to share the screen. Okay.
Anais, can you please allow the presenter to share the screen?
Yeah, can you let me share screen? Yeah, I'm sorry. Yeah, somehow.
I use this opportunity.
I stopped the. Doesn't allow me to share. Okay. All right. Okay, I think now I can. Yeah. All right. Okay. So sorry for the hiccup. Okay. So we have two groups of state-of-the-art string classification method. One is deep learning based, neural network based. The other is the support vector machine based. So deep neural network based models are not really dominating. However, they require a lot of number of training samples. And then the support vector machine. So in this work, we argue support vector machine based string classification are still very good candidate. And then especially when you have smaller training of samples and with a strong theoretical guarantee. All right. Okay, so for the basic idea of string classification is you try to learn two parallel decision boundaries and then to separate the two classes of samples, and so that the distance among the two parallel decision boundaries as far as it can. So if your samples are not separable in the linear space, in the original space, and then there's this basic idea called kernel mapping to map your original space samples into this new space called phi x, so that in the phi x your samples can be separated nicely. And then string kernel exactly is creating this phi x space that separating the strings in the original space that is hard to separate directly, and then into this high dimensional space, and so that we can find two parallel decision boundaries. So the string kernel, like I said, I want to argue string, even though the deep learning based method has been so dominating in almost all the type of prediction tasks. So we found when you have a smaller number of training samples, especially within this like smaller than 8,000 samples for the string classification case, the FastSK has very favorable performance compared to CharCN convolution neural network models, compared to this long LSTM recurrent neural network models, and with much smaller variance. So this figure trying to show you the y-axis is the test AUC, and then the red curve is this really big band from LSTM, and then the orange one is from CharCN, and the FastSK is the blue line. It's actually much better, and with just marginal, almost like negligible variance compared to the other performance. When this task is for a transcription factor binding sites prediction task. All right, so we actually use FastSK and then to test on 3 kinds of string classification tasks. So the first is transcription factor DNA binding site prediction. We're just trying to, when you are given a short segments of DNA genomes, you try to predict, is it a binding site for a specific transcription factor or not? So we can tell for this task, the vocabulary size is 4. So, and there are some like TFs has really smaller number of positive binding sites and like as small as 1,000. So still it's pretty applicable of the string kernel based ideas. So the second string classification task is the protein sequence family prediction task. So it's really from the scope data. So for this task, I mean, the vocabulary size is actually much larger than the DNA case. So sigma equals to 20. And there are some family has a really limited number of training samples as small as 500. So its string kernel based idea is still relevant. And the last, the third kind we actually tried is a language classification task. So like for example, in this case, so can you identify a writer's region just using text info? And then in the paper, we showed a few like other biomedical record-related information extraction tasks. So in total, we tried more than 20 data sets, and they're all for string classification. So, okay, so now let's come back to the basics of string classification. The basic idea of string kernels is just using, represent your strings as a bag of k-mers. So in this example, I show this k, the k-mer just means k continuous substrings in your original string, and then this figure shows k equal to 2. So the string kernel based SVM method, as we reviewed previously, trying to map the string from the k-mer space into from the original string space into this bag-of-Kmer space, so that it's actually easily, you can tell the space is sigma to the k, depends on the dictionary size, and this is really exponential size large. So there's issues with string kernel calculation. It's computationally very expensive. All right, so basics of string kernel. So these as like bag-of-Kmers, I mean, that's if you only use the bag-of-Kmers, that's the basic spectrum kernel. Then the more popular, well-used kernel is called mismatch kernel. So when you're trying to match two sub-Kmers within the string S and the string T, so the matching is not exact, it's not just the exact match. You're also going to consider those mismatch neighborhoods. For example, when I'm considering the mismatch, the neighborhood of AAA, I will also consider AAC, ACA, CA as within this one mismatch distance. So I will consider this as a match when I'm computing the distance among two substrings. So you can actually tell this increased the search of the matching space even larger. And then, because the Kmer representation and the basic spectrum kernel, the search space is sigma to the k. And when let's use an example like the protein case, I mean, your vocabulary size is 20. If your k is equal to 10, and this is like 10 to the 13, like space to represent your basic Kmer space, which is super, super large, and also you're going to get a very sparse representation. And then a state-of-art kernel called gapped Kmer kernel, GKM SVM kernel. And they revised this space representation by promoting an idea called G-Kmer. And then, so in the end, the whole idea is rather than consider all of the possible sigma to the k space, I'm considering only a Gmer space that including my all of the Kmers. So it actually it's more like a wrapper-based idea. So it's makes the space, search space, feature space much smaller than sigma to k. So I will leave details to the paper. Then we follow the GKM SVM formulation, and then I will skip the equations just for the time limit. So in the end, you still consider the matched Kmers, but in the a Gmer space, all possible Gmers in your substrings. So the whole problem converted into you going to count for two strings how many Gmers like size G that matched in the two space with like gaps in this in their definition. So this is a much better reduced feature space representation. However, these papers still use a trie-based data structure, and the computation of this GKM SVM kernel is still pretty slow. So especially when so for example, when you have a little larger like sigma, a larger space like dictionary size, and it's not like possible to extend to the larger sigma case. So we actually convert this GKM SVM into a much simpler like formulation. So I will leave the details to the paper. So in summary, we propose three key strategies to improve the GKM SVM. So we have an even further reduced representation space, and then we provide a direct counting-based method instead of using a trie-based. And then we also further propose a approximation-based algorithm based on our very simple direct counting-based formulation. So I will leave details to the paper. So, and this is the diagram of our algorithm. And the key idea in our method is to just using the sorted array-based data structure. That's the first very big differences versus the GKM SVM. The other is we convert the summation, the kernel calculation into like G-peak-M number of partial kernel profile calculation. And then those, for each of the partial kernel, it's really just a sorted array operation, which is actually super fast. And then further, we propose, we realize you can further approximate that summation of the partial kernels using only some of the kernels, some of the partial kernel, not all of the partial kernels. So in this figure, we actually show only a small portion of the possible mismatch combinations are needed for the strong performance. So when we look at this curve, so this red curve showing the AUC, and then the x-axis shows this number of positions you need to sample. So we call that mismatch combination sampling. And then if in our case, in this figure case, when you have G equal to 10 and M equal to 4, which means you have a Gmer is size 10, you allow 4 mismatch gaps. And then there's 210 possible mismatch partial kernels you need to calculate from the true GChem SVM definition. But we found you really don't need to calculate all the partial kernels. I mean, if only just about less than 5% of the partial kernel got calculated, we can actually tell this red curve, the AUC already start to get converged very nicely. And the accuracy also converged very nicely in the blue curve. We also always use the variance to check when did our kernel like converge. And then the variance is, the orange line is the variance. So the variance got much smaller as we reached the 20% of those mismatch positions when you sample only 20%. But the performance already actually like converge when you have almost only 5%. So this has a huge improvement over our kernel calculation. And then we in the next
1 more minute, just.
Okay, sure. Yeah, sorry. Okay, so this figure trying to show you the figure A shows you the fast SK approximation, and the fast SK exact give you exactly the same, almost the same performance, actually in most cases. So we have only one data set has a slightly worse performance based on that hyperparameter we tune. But all the other data sets, I mean, you only need to sample 5% of the mismatch. And when you have AUC versus the time compared to the GKMS SVM, because GKMS SVM also has a approximation method, but we found that method it's much slower, and also their performance drop huge when you have a larger G. All right, so next is, so we actually show this differences of the computational speed, and we actually much better than GChem SVM. And like in some data sets, like it's about 700 times or faster. In other data sets, it's about like 100 times faster. That's the max speed improvement. If you see the average speed improvement on average, like part of the data set, we got more than 150 times faster. There are some data set we got about like 20 times faster. Then in this figure, and I really need to show is the fast SK predicts really well. So the x-axis is the baseline AUC, the y-axis is the fast SK AUC. And we can actually tell, I mean, versus the GChem SVM versus another baseline GAGO versus LSTM versus CHAR-CNN, we are always better. I mean, we are always like either just a slightly low, but majority is actually much better. So on DNA data, on protein data, also on the NLP test, like NLP data tasks, they are all state-of-the-art baselines we compare. So those points higher than the diagonal lines means we are better. All right, so the last figure, actually last two slides. So we actually got this sensitivity analysis by plotting out when you have a lot of G changes, and then fast SK skips much better compared to the GChem SVM when we have a growing G. Like this is 10 DNA sequence classification task. And also fast SK scales very well with the increasing like parameter M. So this is the gap size, the M. And the last page, I think, okay, so this is the summary. So
I
a fast SKs provides strong prediction performance and scales well with different like dictionary size, scales well with G, scales well with M, and we provide a modern software architecture and also usable toolbox. You can access it from this link. And then there's a bonus property really enabled all of this is we provide a concept through simple kernel algorithms. Sorry for this hiccup and thank you.
Thank you. And June, I'm afraid we don't have time for questions because we are over the time.
Yeah.
And also I should apologize because I missed some of the questions in the previous talks because something is wrong in my screen.
I see.
Probably me.
Yeah, I think it's because of my PDF because
no, it's not your fault. No,
yeah.
I was talking about the questions because I missed some of the questions in the previous. So as I think we had finished, but I remind the speakers that there is a chat, some questions still to be answered. So please go to the chat and see if you can just answer directly there. Thank you, everybody. I place you for the next session at 12 something, 12:10, I think is the
sorry, it's
schedule.
because I shared the PDF, not the PowerPoint. So that's maybe the reason
okay.
of the hiccup.
Don't worry.
Sorry. Thank you. Thank you very much.
We'll have to close the session now. And as I said, we've been placing you for the next part of the session of this data track at 12:10, I think is the original schedule. Perhaps it's delayed, but I'll see you there. Thank you all the speakers. Thank you all the participants for being here. And thank you for the nice presentations. Thank you.